cluster of cells no larger than a grain of sand, a cluster of cells no larger than a grain of rice. A measurement is being taken. Glucose is moving through your blood. The concentration shifts, rises slightly, and a set of cells tucked inside the pancreas registers that shift with a precision that has no equivalent in anything humans have built. Welcome to the Sleepy Biologist. If you've been here before, I'm glad you came back. If this is your first time, you've found a quiet place, and that's enough to know for now. The cells doing the measuring are called beta cells. They sit inside small islands of tissue distributed throughout the pancreas, the islets of Langerhans, and their entire purpose, if purpose is the right word, is to notice, to sense glucose, to respond proportionally, not automatically, not reflexively, but with something that functions like calibration. Here is what actually happens, as best as we understand it. Glucose enters a beta cell through a transporter that doesn't require permission. It simply follows concentration, moving from where there is more to where there is less. Once inside, the glucose is processed. That processing produces ATP, the cell's primary energy currency, and the rise in ATP triggers a cascade of events. A potassium channel closes, the cell's electrical charge shifts, calcium flows inward, and insulin, already packaged and waiting, is released into the blood. The whole sequence takes minutes. In biological time, that is not fast. What's interesting, though not entirely resolved, is that the beta cell doesn't simply switch on at a fixed glucose level. It responds proportionally. A modest rise produces a modest release. A larger rise produces more. The relationship between signal and response is graded, continuous, adjusting in real time to what the blood is actually carrying. There is no single threshold that triggers everything at once. This is worth sitting with for a moment. Your body does not operate on simple on-off logic. Most of its systems don't. The pancreas releases insulin the way a careful person adjusts rather than reacts, not because it is careful in any meaningful sense, but because the biology that shaped it over millions of years settled on graduated responses as more reliable than binary ones. There is a first phase and a second phase to insulin release, and they feel, if feeling's the right word for cellular events, quite different from each other. The first phase is rapid. Within a minute or two of glucose rising, a burst of preformed insulin leaves the beta cells and enters circulation. This burst is short. It peaks quickly and then falls. Researchers believe this initial release serves as a kind of early signal to the liver, a heads-up that something is arriving before the larger wave follows. The second phase is slower. It builds over 20 to 30 minutes and reflects newly synthesized insulin being produced in response to the sustained glucose signal. This phase lasts longer, carries more insulin overall, and is the one most sensitive to damage in the early stages of certain metabolic conditions. It's not entirely clear why the second phase deteriorates first. There are hypotheses, but the mechanism isn't fully settled. The pancreas, during all of this, is also receiving information from elsewhere. The gut, when food is present, releases hormones called incretins, GLP-1 and GIP among them, that reach the beta cells and amplify their response to glucose. The same glucose load, arriving after a meal rather than through an intravenous drip, produces more insulin. The gut is effectively telling the pancreas what is coming before it fully arrives. This is not something most people know their body is doing. It wasn't something researchers fully appreciated until the latter half of the 20th century. The gut communicates with the pancreas. The pancreas adjusts accordingly, the blood carries the result. Your awareness of any of this is zero. You sit or you lie down or you sleep and none of it surfaces. The beta cells measure, the channels open and close, Calcium moves, insulin leaves, the blood carries it outward, towards cells that have been waiting, some more patiently than others, for the signal to arrive. There is something worth noticing in how quietly this runs. Not dramatic, not urgent. Just a series of chemical events, each triggering the next, each proportional to what came before. The pancreas finishes its calculation long before you notice anything. And the insulin, now in circulation, begins the slower work of finding where it is needed most, and what each cell in its own way is prepared to do with it. You have roughly 37 trillion cells in your body, and right now not one of them is asking for the same thing. That number is hard to hold. 37 trillion. If you tried to count them one per second without stopping, you would still be counting long after every person alive today had lived and died. And yet somehow the body keeps track, not of each cell individually, but of what each tissue type needs, when it needs it, and how urgently. It does this continuously, without error accumulating fast enough to matter, without a central room where decisions get made. Insulin enters this situation like a signal broadcast across an enormous and varied crowd. Some cells hear it immediately, some are slower to respond, and some, this is the part that stays with you, are built to respond to insulin in ways that serve a purpose entirely different from their neighbours, even when the molecule arriving at their surface is chemically identical. Your muscle cells want glucose for movement, not movement happening right now necessarily, but potential movement, the kind the body is always quietly preparing for. Skeletal muscle carries insulin receptors on its surface, and when insulin binds, a cascade of internal events eventually brings glucose transporters, GLUT4 specifically, up from inside the cell to the membrane. They migrate outward, they dock, and glucose following its gradient enters. This migration is not instantaneous. The transporter has to travel from where it was stored, inside small vesicles in the cell's interior, all the way to the outer membrane. That journey takes time. It involves signaling proteins, phosphorylation events, cytoskeletal rearrangements, a whole quiet infrastructure of movement that has to activate before a single glucose molecule crosses over. You don't feel any of this, but it is happening in you right now, or it was an hour ago, or it will be in 20 minutes. The timing depends on when you last ate, what you ate, how your body is weighted at this particular moment toward rest or readiness. Fat cells respond to insulin too, but their response feels, if cellular behavior can feel like anything, more absolute. Adipose tissue is extraordinarily sensitive to insulin. A relatively small rise in the hormone is enough to halt fat breakdown almost entirely. The enzyme responsible for releasing fatty acids from stored fat, hormone-sensitive lipase, becomes inhibited. Fat cells stop releasing, they shift instead towards storage. This happens early before muscle cells have fully committed to their own response, and it happens at lower insulin concentrations than most other tissues require. Why so sensitive? There's something almost protective in it. The body, when food is present and insulin is rising, wants to preserve what it has recently stored. Fat tissue reads the insulin signal and essentially pauses its outflow. It doesn't need much convincing. Liver cells occupy a different position entirely. The liver receives insulin before almost any other organ because of where it sits in relation to the portal vein, the vessel that drains blood from the gut directly into the liver before it circulates anywhere else. This means the liver sees the first wave of insulin released after a meal, at concentrations that are considerably higher than what the rest of the body eventually receives. And the liver does not simply comply. It listens to the insulin signal, but it weighs it against its own internal state. How much glycogen is already stored? What is the current blood glucose doing? Has the liver been releasing glucose for the last several hours while the body was fasting? These are not conscious calculations. They are the result of enzyme states, protein modifications, and feedback signals that collectively produce a response that is, at best, conditional. Sometimes the liver agrees fully. It stops releasing glucose, begins storing it as glycogen, and shifts toward the kind of metabolic behavior insulin is requesting. Sometimes it only partially agrees. It reduces its glucose output without stopping entirely, continuing a slow release even as insulin climbs. Researchers still find this interesting. The liver's partial compliance is not a failure of signaling exactly. It reflects the liver's role as the body's central metabolic negotiator, always balancing what the blood is asking for against what the liver believes the situation actually requires. There is something almost familiar in that. The organ that keeps the most information is also the one least willing to be fully directed by a single signal. Across all these tissues, muscle waiting for glucose, fat cells pausing their release, the liver calculating its own position, insulin is doing the same thing, binding the same receptor type, triggering the same fundamental pathway, and yet the outcome in each place is different, shaped by what the cell was built to do, what it has been doing recently, and how urgently it has been waiting. The body doesn't send different versions of insulin to different places, it sends one signal, and each tissue, in its own way, decides what that signal means. There is no map for where insulin goes first. No routing system, no address attached to each molecule as it leaves the pancreas and enters the portal vein, no instruction embedded in its structure that tells it which tissue to seek before the others. It moves the way everything dissolved in blood moves, carried by flow, distributed by pressure, delivered according to the architecture of vessels rather than any hierarchy of need. And yet, despite the absence of deliberate ordering, something that functions like a queue emerges anyway, shaped entirely by the body's own geography. The liver, as noted, receives the signal earliest, not because it is most important, but because it is closest to the source, positioned at the end of a vascular route that drains the gut and carries the first wave of post-meal insulin directly into hepatic tissue before the rest of the circulation has seen much of anything. This proximity is not incidental, it is structural, the result of an arrangement that has persisted across vertebrate evolution, suggesting that having the liver intercept insulin early confers some advantage the body has never found reason to abandon. By the time insulin reaches the heart and enters the general circulation, the liver has already begun its response, already begun adjusting its glucose output, already started the cascade of enzyme changes that will define its behavior for the next hour or more. From the heart, insulin disperses. It enters arteries, moves into arterioles, and eventually reaches the capillary beds that supply individual tissues. And here the cue becomes less about sequence and more about local conditions, blood flow, capillary density, the metabolic state of the tissue at that particular moment. Skeletal muscle, which makes up a substantial portion of total body mass and accounts for a large share of post-meal glucose uptake, receives insulin through capillaries that are not all open simultaneously. At rest, a significant fraction of muscle capillaries are closed, and insulin's own presence, along with other signals, contributes to their gradual recruitment, a process called capillary recruitment, which increases the surface area available for insulin and glucose to cross from blood into tissue. This recruitment takes time, unfolding over minutes rather than seconds, and it means that even after insulin has arrived in the blood supply near a muscle, the actual delivery of that insulin to muscle cells is still in progress, still expanding, still opening new territory as the signal strengthens and the vessels respond. What stands out most in considering this is how much of the body's coordination happens at cells most people never think about, Not organ to organ, not even cell to cell, but at the level of individual capillary segments deciding whether to carry flow or remain quiet. Adipose tissue receives insulin through its own capillary networks, and because fat cells respond at lower insulin concentrations than muscle, they often reach their full response earlier in the post-meal window, even though the insulin arriving at their surface comes from the same circulation that is simultaneously supplying everywhere else. The difference is sensitivity. Fat cells simply require less of the signal to commit fully to their response, which means they are effectively prioritized not by the body's routing but by their own receptiveness, their own threshold for action set lower than the threshold of other tissues. The brain, during all of this, sits apart. Most neurons obtain glucose through transporters that do not depend on insulin, which means the brain is not waiting in any queue at all. It draws from the blood continuously, independently, without requiring the signal that is currently organizing the behavior of nearly every other major tissue. There is something worth pausing on in that arrangement. The quiet separation of the organ that processes everything from the system that is managing everything else. The brain receiving its fuel on entirely different terms, as though the body recognized early that one system needed to remain insulated from the fluctuations that govern the rest. And peripheral tissues, the skin, the connective tissue, the slower and less metabolically urgent structures, receive insulin last. Not because the circulation forgets them, but because their demand is lower, their capillary density thinner, their response to insulin less consequential in the immediate window following a meal. They are part of the queue, but they sit toward its end, receiving what remains after the liver and muscle and fat have taken what the signal required them to take. The queue then is not a line. It is a pressure gradient, a sensitivity gradient, a geography of vessels and thresholds and competing demands that together produce something resembling order without anyone having designed the order in advance. Insulin moves through it continuously, binding where it can, triggering what it reaches, adjusting a system that is always, in every tissue simultaneously, in the middle of responding to something. By the time the last capillary beds have received their share of the signal, the pancreas has already begun reading the result, watching glucose fall, sensing the shift, preparing to reduce its output in proportion to how well the distribution has worked. The queue resolves, and somewhere in the pancreas, the measurement begins again. Something is waiting at the edge of the cell, not pressing, not urgent, just present, suspended in the fluid just outside the membrane, held in concentration, tilting gently toward the inside where there is less of it. Glucose moves down gradients, always has. It doesn't push, it doesn't signal ahead, it simply goes where there is room for it, and waits at boundaries where the room hasn't opened yet. The boundary here is a lipid membrane. 2 layers of fat molecules arranged tail to tail, forming a barrier that glucose, despite being small by molecular standards, cannot cross on its own. The membrane is selective in a way that feels almost deliberate, though there is nothing deliberate about it. It is just chemistry. Hydrophilic things don't pass through hydrophobic barriers without help. The help is a protein, specifically a glucose transporter, a transmembrane protein that spans the full width of the membrane with a channel running through its center that can open on one side, accept a glucose molecule, and deposit it on the other. The transporter doesn't consume energies to do this. It uses the gradient itself. The glucose arrives from where it is more concentrated, passes through, and emerges where it is less so. Passive, quiet, entirely dependent on the difference existing in the first place. There are several types of these transporters, and this is where it becomes interesting. GLUT1 is present in nearly every cell in the body, It runs continuously, requiring no additional signal, maintaining a baseline uptake that keeps cells supplied during ordinary circumstances. It is not waiting for insulin. It was never waiting for insulin. It simply sits in the membrane and does its work regardless of what the hormone situation happens to be. GLUT4 is different. GLUT4 spends most of its time inside the cell, tucked into small membrane-bound compartments called vesicles, stored in the cell's interior, nowhere near the surface where glucose is waiting. In this state, the transporter is functionally invisible. Glucose outside cannot reach it. The gradient exists, but the path is closed. Insulin is the reason the path opens. When insulin binds its receptor on the surface of a muscle or fat cell, a cascade begins, a sequence of phosphorylation events, protein activations, signals moving inward from the membrane toward the cell's interior. One of the things this cascade eventually does is trigger the vesicles carrying GLUT4 to move toward the surface. They travel along cytoskeletal tracks. They reach the membrane. They fuse with it, and the transporters, now embedded in the outer membrane, become accessible. Glucose enters. This is the part that tends to get compressed in the textbook descriptions, the idea that insulin allows glucose into cells, as though insulin itself were carrying something, as though it were opening a door it had a key to. It doesn't carry anything. It doesn't touch the glucose. It binds its receptor, triggers a chain of intracellular events, and those events move the transporter to where the glucose can use it. The glucose still moves on its own terms, down its gradient, through the transporter, into the cell. Insulin changed the number of available gates, that's all. And yet that change, the difference between a membrane with few GLUT4 transporters and one suddenly populated with them, determines whether the cell fills or stays waiting, whether muscle can replenish its glycogen, whether fat tissue can take up fuel for storage, whether blood glucose begins to fall after a meal or continues rising. The gate was always there, stored, ready, waiting for the signal that would bring it to the surface. You start to notice, sitting with this, that a great deal of what insulin does is less about creation and more about relocation, moving things that already exist to places where they can be used, bringing the transporter up, repositioning enzymes, shifting the location of proteins that were already synthesized, already functional, just not yet at the surface where the work needed doing. The cell had the equipment all along. Insulin changed where the equipment was standing. Once GLUT4 is at the membrane, glucose enters for as long as the gradient supports it, until the inside concentration approaches the outside concentration, or until the signal fades and the transporters are slowly retrieved back into the cell's interior, recycled into vesicles, waiting again. The retrieval is gradual. The transporters don't disappear the moment insulin begins to fall. They linger at the surface a little past the signal's peak, continuing to admit glucose while the hormone is already declining elsewhere. The cell's response outlasts the signal by some margin, not indefinitely, not forever, but long enough that the boundary between insulin's presence and insulin's absence is blurred at the membrane level. The gate stays open a little after the signal is gone. Then slowly, quietly it returns to where it came from, and the glucose that made it through, already inside, already being processed, moves deeper into the cell's chemistry, becoming energy, becoming storage, becoming part of the ongoing work the cell was doing before any of this began, and will continue doing long after the transporter has been retrieved and the membrane has returned to its quieter state. Something is still moving at the edge of the cell. Less of it now, the gradient narrowing. The work of this particular wave of insulin approaching its end, though the effects of it, the glycogen being built, the enzymes still running at their activated rates, will persist for a while yet, carrying the signal's meaning forward into time even after the signal itself has faded. The liver has been managing glucose longer than any other organ in your body has been doing anything. Not your liver specifically, but the liver as a structure, as an evolutionary solution to the problem of fuel storage and release, predates most of what we think of as complex animal physiology. It was handling glucose metabolism in early vertebrates long before the endocrine system had developed the sophistication it carries now. By the time insulin arrived as a regulatory signal, the liver already had its own logic, its own priorities, its own way of deciding what the body needed. Insulin, in some respects, learned to negotiate with a system that was already ancient. Your liver sits just beneath your diaphragm on the right side, and it receives blood from two directions simultaneously. The hepatic artery brings oxygenated blood from the heart. The portal vein brings nutrient-rich blood directly from the gut, blood that has passed through the intestinal wall and collected everything absorbed from a recent meal, including the glucose that triggered the insulin release that is currently in progress. This dual supply matters. The insulin arriving via the portal vein is considerably more concentrated than the insulin that eventually reaches your arm, your legs, your skin. The liver intercepts a large fraction of it. Some estimates suggest more than half of the insulin secreted after a meal is cleared by the liver before it enters general circulation. The liver is not just a recipient of the signal, it is in part a regulator of how much of that signal the rest of the body receives. I tend to think this arrangement is underappreciated. Most descriptions of insulin focus on muscle and fat, on glucose uptake, on the transporter story. The liver's role as both target and filter sits somewhat quietly in the background, doing more than the simplified version suggests. What insulin asks the liver to do, when it arrives in sufficient concentration, is to stop releasing glucose and start storing it. Between meals, the liver has been doing the opposite. It breaks down glycogen, the branched glucose polymer stored in its cells, and releases glucose into the blood to maintain circulating levels during the fasting interval. It also produces glucose from non-sugar precursors, a process called gluconeogenesis, drawing on amino acids and glycerol to synthesize new glucose when stored glycogen runs low. These two processes, glycogenolysis and gluconeogenesis, are the liver's contribution to keeping blood glucose stable during the hours when no food is arriving. Insulin suppresses both. It activates an enzyme called phosphodiesterase, which lowers levels of cyclic AMP inside liver cells, which in turn reduces the activity of the enzymes driving glycogen breakdown. It promotes the activity of glycogen synthase, shifting the liver towards storage rather than release. It inhibits the transcription of genes encoding gluconeogenic enzymes, reducing the liver's capacity to produce new glucose over longer timescales. This is not a single switch. It is a reconfiguration across multiple levels, enzymatic, signaling, genetic, each operating on a different timescale, each contributing to a shift in the liver's overall metabolic posture. And yet the liver does not always comply fully. In the early stages of insulin resistance, the liver shows a specific and somewhat peculiar pattern. It stops suppressing gluconeogenesis in response to insulin, while still responding to insulin signals to promote fat synthesis. The two processes, glucose production and lipid synthesis, become uncoupled in a way that doesn't make obvious sense from the outside. The liver continues making glucose when insulin says not to, while simultaneously making fat in response to the same signal. Researchers have spent considerable effort trying to understand this selective resistance. It's not entirely resolved. The leading hypothesis involves the branching of insulin's intracellular signaling pathway, different downstream effects using different molecular routes, each with different sensitivity to disruption. One branch stays responsive, the other becomes resistant. The result is a liver that is partially listening and partially not, in a pattern that happens to be particularly unfavourable for metabolic health. I find it genuinely interesting that the same signal can be heard differently within a single cell depending on which pathway it travels. It suggests that insulin resistance is not a simple volume problem, not just too little signal reaching the liver, but something more like a routing problem, where parts of the message arrive intact and others don't. The liver, for its part, continues operating. It processes the blood arriving from the gut. It filters the insulin. It responds where its signaling is intact and continues its old behavior where it isn't. It stores some glucose, it releases some. It synthesizes fat when the carbon load exceeds what glycogen can accommodate. It does all of this simultaneously, running dozens of metabolic pathways in parallel across billions of hepatocytes, each cell making its own local adjustments based on the concentrations of signals it happens to be receiving at that moment. From the outside, from the blood's perspective, what emerges is a net output, a number. How much glucose the liver is adding to the circulation per minute or subtracting from it. That number shifts over the course of hours, responding to insulin, to glucagon, to the liver's own stored fuel levels, to signals arriving from fat tissue and muscle and the nervous system. The liver considers all of it. It has been doing this for a very long time, and when insulin arrives after a meal, the liver doesn't simply receive the signal and obey. It weighs it. It integrates it against everything else it currently knows, and then it responds in the way that its present state, its enzyme activities, its glycogen levels, its recent history, determines is appropriate. Sometimes that response is exactly what insulin requested. Sometimes it is close, and the insulin, having done what it can at the liver, moves on into the wider circulation, carrying its signal outward toward tissues that have been waiting with varying degrees of patience for their turn to respond. It doesn't begin as a refusal. Nothing dramatic happens, no signal breaks, no receptor disappears overnight. The cell doesn't decide anything. It simply begins at some point that cannot be precisely located to respond a little more slowly than it once did. You wouldn't notice it from the outside. The blood glucose numbers might still look acceptable. The pancreas is compensating, producing more insulin than it once needed to produce in order to achieve the same result. The system is still working. It has just started working harder to stay in the same place. This is how insulin resistance begins in most people, not with a threshold crossed, but with a gradual drift, a delay here, a slightly blunted response there, the cell receiving the signal and to doing less with it than it previously would have. The receptor itself is usually intact. Insulin still binds. The binding affinity, the strength of the connection between insulin and its receptor, doesn't change dramatically in the early stages of resistance. What changes is what happens after binding. The signal that should travel inward from the receptor, activating a cascade of phosphorylation events that eventually moves GLUT4 transporters to the surface, begins to lose something in transmission. The key disruption involves a protein called IRS1, insulin receptor substrate 1. When insulin binds its receptor, the receptor phosphorylates IRS1, and this phosphorylated form then activates downstream signaling. In a resistant cell, IRS1 arrives at that step already carrying inhibitory phosphorylation on different sites, placed there by other kinases, activated by inflammation, by excess fatty acids, by cellular stress signals that have been accumulating quietly. The receptor fires, IRS1 is present. But the message doesn't carry the way it should. What causes those inhibitory signals to accumulate in the first place is not a single thing. excess lipid inside muscle cells, diacylglycerol specifically, activates a kinase called PCK-theta, which phosphorylates IRS1 on serine residues rather than tyrosine residues, and serine phosphorylation at those sites inhibits rather than activates downstream signaling. Inflammation does something similar through a different kinase. Mitochondrial stress contributes. Endoplasmic reticulum stress contributes. Several processes converge on the same point, and the result, from the cell's functional perspective, is that the GLUT4 vesicles don't migrate as efficiently. Fewer transporters reach the surface per unit of insulin. Glucose uptake slows. The cell continues doing most of what it does: protein synthesis, energy production, maintenance, but its response to the hormone has become attenuated in a way that takes time to become visible at the level of blood glucose. The pancreas notices before you do. Beta cells are sensitive to blood glucose in a way that makes them early detectors of resistance. When muscle and fat cells take up less glucose in response to insulin, blood glucose stays elevated slightly longer after meals. The beta cells sense this and respond by releasing more insulin during the second phase of secretion. For a while, sometimes years, this compensation is sufficient. Blood glucose remains in an acceptable range because the pancreas is producing enough extra insulin to overcome the blunted cellular response. This compensation has a cost that accumulates slowly. Beta cells working at chronically elevated output are under sustained metabolic stress. They are producing and releasing more insulin than they were built to sustain indefinitely. Over time, some beta cells begin to lose function. A small fraction undergo apoptosis. The total secretory capacity of the pancreas quietly declines. There is something uncomfortable in this sequence. The idea that the compensation itself contributes to the eventual failure of compensation. The body's solution to the problem participates in the problem's progression. It's not entirely clear at what point the balance tips, or whether it tips at the same rate in different people, or what determines who compensates successfully for decades and who doesn't. There are genetic factors. There are factors related to how much lipid accumulates in muscle and liver. There are factors related to the degree and duration of inflammation. The picture is real but still partially unresolved. Meanwhile, the cells continue their slow drift. In fat tissue, insulin resistance affects not just glucose uptake but also the suppression of lipolysis, the breakdown of stored fat. A resistant fat cell requires more insulin to suppress lipolysis than it once did. Between meals, when insulin is low, this might not matter. But in the post-meal period, when insulin is rising but fat cells are less responsive, lipolysis continues at a higher rate than it should. More fatty acids enter the blood. Some of those fatty acids reach the liver, some reach the muscle, and there they can contribute to the very intracellular lipid accumulation that drives further insulin resistance. The loop runs quietly, each component contributing to the conditions that sustain the others. No single cause, no moment when everything changes. Just a gradual shift in the balance between signal and response, spreading across tissues over time, advancing most quickly in the places where the conditions for it are most favorable. The cell hasn't refused. It has simply stopped hearing as clearly as it once did. And somewhere still the pancreas is measuring, noticing the gap between what it sent and what the blood is showing, and preparing as it had been preparing for some time now to send a little more. You've probably felt this before without having a name for it. The heaviness that arrives after a large meal, the way the body seems to settle inward, shifting into a different mode, slower and more contained than it was an hour ago. Some of that feeling has a biological source, and some of it originates specifically in what fat tissue is doing, or more precisely, in what it has just been told to stop doing. Fat cells are not passive storage. They are metabolically active, continuously releasing and reclaiming fatty acids in a cycle that never fully stops. Even now, at rest, your adipose tissue is in motion at the molecular level. Lipase enzymes breaking down stored triglycerides, fatty acids leaving through the cell membrane, entering the blood, traveling toward tissues that can oxidize them for fuel. This process runs as a background operation during fasting and between meals, and it provides a significant fraction of the energy your heart and resting muscle and other tissues use when glucose is scarce. Insulin interrupts this, not gently, not gradually. Adipose tissue is exquisitely sensitive to insulin, more sensitive than muscle, more sensitive than most other tissues in the body. And when insulin rises after a meal, fat cells respond to concentrations that would barely register as a signal elsewhere. The enzyme at the center of this response is hormone-sensitive lipase, and insulin's effect on it is inhibitory. The enzyme slows. The breakdown of stored triglycerides slows with it. The outflow of fatty acids into the blood narrows, then nearly stops. The fat cell, in a sense, closes. What makes this sensitivity striking is its asymmetry. It takes relatively little insulin to suppress lipolysis almost completely, but it takes considerably more insulin, higher concentrations sustained longer, to drive meaningful glucose uptake into fat cells via GLUT4. The two responses have different thresholds. Stopping the release comes first. Increasing the intake comes later, at higher signal levels. This ordering has a logic to it. When food has just been eaten and glucose is rising, the body's first priority is to stop fat from competing as a fuel source. Muscle and other tissues will be receiving glucose shortly. Having fatty acids simultaneously flooding the bloodstream would create a kind of metabolic crowding, tissues receiving energy signals from two directions at once, with all the regulatory complexity that entails. By shutting down fat release early, at low insulin concentrations, the body clears the field for glucose to take over as the dominant fuel. The fat cell waits. It holds what it has. Its lipase enzymes sit inhibited. The triglycerides that were in the process of being broken down remain as they are. The fatty acids that had been cued for release stay inside. This is the waiting that fat tissue does, not passive, not inert, but held. Suspended in a state of arrested release while the rest of the metabolic event plays out around it. Insulin also acts on another enzyme in fat cells, lipoprotein lipase, which sits on the surface of capillaries supplying adipose tissue and breaks down triglycerides carried in the blood by lipoproteins, releasing fatty acids that the fat cell can then absorb and store. Insulin activates this enzyme, so at the same time that hormone-sensitive lipase is being inhibited, reducing outflow, lipoprotein lipase is being promoted, increasing the capacity for inflow. The fat cell is simultaneously less likely to release and more capable of absorbing. The net effect is a tissue that is, during the post-meal insulin period, oriented almost entirely towards storage. You already know this feeling in some form. The body after a meal feels different from the body before one. Some of that difference is gastric, the physical presence of food, the stretch of the stomach wall, the release of gut hormones. But some of it is this: adipose tissue shifting its entire orientation from a tissue that was quietly releasing to one that is quietly accumulating, holding more than it is giving, waiting for the insulin signal to pass before returning to its default behavior. When insulin eventually falls, hours after the meal, as glucose has been taken up and the pancreas has reduced its output, the fat cell begins to re-emerge from its suspended state. Hormone-sensitive lipase reactivates. The inhibition lifts. Fatty acids begin leaving again, slowly at first, then more steadily as insulin continues to fall. The tissue returns to its background operation. The cycle that was interrupted resumes, and the fatty acids that flow back out into the circulation, released now during the post-absorptive period, become the fuel that carries the body through the hours until the next meal arrives and insulin rises again, and fat tissue, responsive as ever to the smallest increase in the signal, closes once more. There is something in this rhythm, the opening and closing of fat tissue around meals, the sensitivity that makes it the first responder and the last to fully reopen. That reflects how central adipose tissue is to the body's overall fuel economy. Not a warehouse that fills and empties on command, but a participant with its own timing, its own thresholds, its own way of reading the signal that everyone else is reading and deciding earlier than most what the moment requires. The waiting is the work, and the fat cell, more than almost any other tissue, has made waiting into something precise. The brain never waits for permission. While the rest of the body adjusts its glucose uptake according to insulin, while muscle cells hold their GLUT 4 transporters in reserve, while fat tissue suspends its release, while the liver recalculates, the brain draws from the blood continuously without pausing, without requiring any signal to authorize the transaction. This is not an oversight in the system. It is the system's most carefully preserved arrangement. The neurons that make up most of your brain's working tissue rely on glucose as their primary fuel, and they consume it at a rate that is, relative to the brain's size, remarkable. The brain accounts for roughly 2% of body mass, but uses somewhere around 20% of the body's resting energy. That consumption runs day and night, whether you are solving problems or lying still in the dark, whether you have eaten recently or not. It does not throttle down in proportion to what the rest of the body is doing. The transporters responsible for this are primarily GLUT1 and GLUT3. GLUT1 lines the blood-brain barrier, the specialized capillary endothelium that separates the brain's fluid environment from general circulation. It moves glucose across this barrier continuously, driven by the concentration gradient, requiring no insulin to function. GLUT3 sits on the surface of neurons themselves, and it has a particularly high affinity for glucose, meaning it can extract glucose from the surrounding fluid even when concentrations are relatively low, binding its substrate more tightly than the transporters used by muscle or fat. The neuron, in other words, is built to take what it needs before it runs short. There is an insulin receptor in the brain. This is worth pausing on because it complicates the simple story of brain independence. Insulin does cross the blood-brain barrier, transported by a receptor-mediated mechanism, and insulin receptors are present in the hypothalamus, the hippocampus, the cortex, regions involved in appetite regulation, memory, and cognition. Insulin in the brain appears to influence hunger signaling, synaptic plasticity, and the regulation of body weight. It is doing something there, even if glucose uptake in neurons is not primarily what it is doing. The brain's relationship with insulin is, this feels like the right word, selective. It accepts the signal for certain purposes. It ignores it for others. The fundamental business of fueling neurons proceeds independently. The subtler work of appetite and plasticity involves insulin in ways that researchers are still tracing. What the exemption means practically is that during hypoglycemia, when blood glucose falls to genuinely low levels, the brain does not have a backup fuel reserve the way muscle does. Muscle stores glycogen and can draw on it locally. The brain stores almost none. It depends on a continuous supply from the blood, and when that supply is interrupted, the consequences arrive quickly and in a particular order. Confusion first, then difficulty concentrating, then more serious impairment, then loss of consciousness if the drop is severe enough. The brain's priority status, its insulin-independent access, Its high affinity transporters, its continuous demand, is also its vulnerability. The arrangement that protects it under normal conditions offers no buffer when the supply itself fails. This is something you might notice in yourself on a long morning without food. The particular quality of that mental dimming, the way thinking becomes effortful, the way attention slides, is partly the brain approaching the lower edge of its comfortable glucose range. Not dangerous, not even close to the levels that cause real impairment, but perceptible in the way that only things happening inside the organ doing the perceiving can be. The brain noticing its own fuel state. Somewhere in the hypothalamus, glucose-sensing neurons, cells that are specifically responsive to changes in glucose concentration, are registering the shift and beginning to generate the signals that will eventually surface as hunger. The feeling of needing to eat is, in part, the brain monitoring its own supply and initiating the behavior that will replenish it. The organ that is exempt from waiting is also the organ that decides when waiting has gone on long enough. It monitors the blood, it reads the concentration, it sends the signal that brings you to a kitchen or a table or a piece of fruit left on a counter. And when glucose arrives and insulin rises and every other tissue in the body adjusts its behavior in response to the signal, the brain continues as it was, drawing from the blood at its own rate, through its own transporters, on its own terms. The exemption holds. The brain never waited, and it is already quietly deciding when it will need to ask again. There is a version of your body that handles glucose well in the morning, and a version that handles it less well at night. And they are the same body running the same biology, separated only by the position of the clock. This is not a small difference. Studies measuring glucose responses to identical meals eaten at different times of day have found meaningful variation. The same food, the same person, the same quantity, producing a higher and more prolonged blood glucose response in the evening than in the morning. The insulin response differs too. Not because the pancreas is failing at night exactly, but because the tissues receiving the signal are less prepared to act on it. The reason for this reaches back further than any individual meal or any single night's pattern of eating. It reaches into the architecture of the cell itself, into a set of genes that have been running a 24-hour oscillation since before complex animals existed, the circadian clock, which operates not just in the brain but in nearly every cell of the body, including the beta cells of the pancreas and the muscle and fat cells that insulin will eventually address. Each of these cells contains a molecular clock, a feedback loop built from interlocking proteins, some of which activate genes and some of which inhibit them, cycling through a full period in approximately 24 hours. The proteins clock and BMAL1 form the positive arm of this loop, driving expression of genes including PER and CRY, whose protein products accumulate and eventually inhibit SLOC and BMAL1, reducing their own production in a self-limiting cycle that resets each day, entrained to light and feeding signals arriving from the environment. This is ancient machinery, deeply conserved. What the circadian clock does to insulin sensitivity is, I tend to think, one of the more underappreciated aspects of metabolic biology. The clock controls the expression of GLUT4 in muscle cells, the transporter that moves to the surface in response to insulin, so that GLUT4 expression peaks during the active phase and falls during rest. It controls the expression of insulin receptor substrate proteins, the signaling intermediaries whose disruption underlies insulin resistance. It regulates the activity of enzymes in the liver that determine whether incoming glucose is stored or processed. It influences the timing of insulin secretion from the pancreas itself, shaping not just how much insulin is released, but when the release is most robust. The result is a system whose sensitivity to insulin rises and falls across the day in a pattern that, in most people, peaks in the morning and early afternoon and declines toward evening and night. Eating late, then, means eating into a period of reduced readiness. The glucose arrives, the pancreas responds, but the downstream tissues, the muscle, the fat, the liver, are operating in a phase where their molecular machinery is less configured for rapid glucose uptake, where the transporters are less abundant at the surface, where the signaling intermediaries are expressed at lower levels. The same insulin signal achieves less than it would have achieved hours earlier. It accumulates more slowly than it clears. The blood glucose stays elevated longer. There is something almost wistful about this. If biology can produce that feeling, the idea that the body has an opinion about when food should arrive, an internal expectation shaped over evolutionary time by patterns of light and activity and feeding that no longer map cleanly onto how most people actually live. The circadian system was tuned for a world where activity and eating clustered in the light hours and rest and fasting clustered in the dark. Modern schedules distribute eating across a much wider window, and the molecular clocks in metabolic tissues are running their program regardless, expressing their transporters and enzymes on their own schedule, available or not available, according to a timing that has nothing to do with when dinner happens to be ready. Disrupted circadian rhythms, from shift work, from irregular sleep, from social jet lag, are associated with metabolic changes that look in many respects like mild insulin resistance. The association is not merely correlational. Experimental studies deliberately shifting the timing of sleep and meals in otherwise healthy people have produced measurable increases in post-meal glucose and decreases in insulin sensitivity within days. The molecular mechanism runs through the same clock genes, the same transporter expression patterns, the same cascading effects on signaling intermediaries. The timing matters as much as the content. A diet assessed only by its macronutrient composition, without regard for when those macronutrients arrive relative to the body's circadian phase, is missing something real. Not something that overrides everything else. Not something that makes late eating catastrophic for a healthy person, but something that the biology registers and accumulates and responds to over time in ways that eventually become visible. The cellular memory of timing is real. The muscle cell that handles glucose at 8 in the morning and the muscle cell that handles glucose at 11 at night carry the same genome, run the same insulin signaling pathway, use the same transporter. But the proteins currently expressed, the enzymes currently active, the channels currently configured, these reflect a history of timing, a pattern of when activity has occurred relative to light and dark, a circadian context that shapes the cell's present capacity. The body remembers what time it usually is, and it prepares accordingly, whether or not the preparation matches what is actually coming. Insulin does not vanish when eating stops, it falls. Gradually, over the course of an hour or more, the concentration in the blood declines as the pancreas reduces its output and the liver continues clearing the hormone from circulation. The decline is not a cliff, it is a slope, and different tissues respond to it at different rates, withdrawing from their post-meal posture incrementally, each according to its own sensitivity to the falling signal. Fat tissue responds first. The threshold at which insulin suppresses lipolysis is low, which means fat cells were the first to close when insulin rose, and they are among the first to reopen when it falls. As the concentration drops below the level required to hold hormone-sensitive lipase inhibited, the enzyme begins to reactivate. Slowly at first, a partial disinhibition, a gradual resumption of triglyceride breakdown. Fatty acids begin leaving the fat cell again, entering the blood in increasing amounts, becoming available to the tissues that will shift toward oxidizing them as glucose availability declines. this transition is not announced. There is no signal that says the post-meal phase has ended and the fasting phase has begun. The two states blend into each other across a window of time that varies depending on meal size, composition, and individual metabolic rate. What changes is a set of concentrations: insulin falling, glucagon beginning to rise, fatty acids increasing in the blood, glucose declining slowly toward its fasting baseline. And those concentrations collectively shift the behavior of every metabolic tissue in the body. The liver reads this shift early. As insulin falls and glucagon rises, the liver begins transitioning back toward glucose output. The enzymes that insulin had suppressed, the ones driving glycogen breakdown and gluconeogenesis, begin reactivating. Glycogen phosphorylase, which cleaves glucose units from stored glycogen, becomes active. The liver starts releasing glucose back into the blood, replacing what peripheral tissues are consuming, maintaining the circulating level within a range that the brain and other insulin-independent tissues can continue drawing from without interruption. This is one of the things the liver does that tends to get underweighted in simplified accounts of glucose regulation. The continuous active maintenance of blood glucose during fasting, not as a passive default, but as a regulated output that responds in real time to signals arriving from the pancreas, from the blood, from the nervous system. The liver is not simply releasing glucose because nothing is stopping it. It is releasing glucose because glucagon is telling it to, because the glucose concentration it is monitoring has dropped to a level that warrants a response, because its glycogen stores are sufficient to support that response at the current rate. Muscle during this same interval shifts its fuel sourcing. In the hours after a meal, as insulin falls and fatty acid availability increases, skeletal muscle progressively reduces its glucose oxidation and increases its reliance on fat. This shift is driven partly by the falling insulin signal. Fewer GLIT4 transporters at the surface, less glucose entering, and partly by the increasing concentration of fatty acids in the blood, which muscle cells take up and oxidize in preference to glucose when both are available and insulin is low. The Randall cycle, as this competition between fuels is sometimes called, operates continuously in muscle, with the balance tilting toward fat during fasting and toward glucose after meals. The GLUT4 transporters that migrated to the muscle cell surface in response to insulin are slowly retrieved. They return to their intracellular vesicles. The surface density of transporters falls. Glucose entry into muscle slows, not abruptly but progressively, tracking the decline in insulin that is itself tracking the decline in blood glucose that followed the meal. What stands out in observing this sequence is how much of what the body does between meals is active rather than passive. The fasting state is not the body doing nothing while it waits for the next meal. It is a distinct metabolic configuration: fat releasing, liver outputting, muscle shifting fuels, glucagon coordinating, insulin low but not absent, that requires continuous regulation to maintain. The organs are working, the hormones are signaling, the enzymes are running. The body between meals is not resting from metabolism, it is running a different version of it. And across the hours of this interval, whether it lasts 3 hours or 13, the tissues are monitoring the same parameters they always monitor, adjusting their outputs, reading the concentrations in the blood, responding to what those concentrations say about the current state of fuel availability. Fat tissue releases a little more as the fast extends. The liver draws down its glycogen, and if the fast continues long enough, begins increasing gluconeogenesis to compensate for the declining glycogen stores, synthesizing glucose from amino acids and glycerol to maintain what the blood requires. Muscle continues shifting toward fat oxidation, sparing glucose for tissues that cannot make the switch. The brain draws steadily, as it always does, from whatever the blood is carrying, and somewhere in the pancreas, the beta cells that were active after the last meal have reduced their output to a basal rate, a quiet, low-level insulin release that continues even during fasting. Not enough to drive significant glucose uptake into muscle or fat, but enough to prevent the liver from releasing glucose at an unregulated rate. The interval between meals holds its own balance. It is not waiting. It is a state the body knows how to run, has always known how to run, and returns to with the same quiet competence it brings to everything else, each time the last meal recedes and the next one has not yet arrived. There is a point past which waiting changes something, not a precise point, not a threshold that appears on any chart or gets recorded in any clinical measurement, just a place in time somewhere during a prolonged deprivation of glucose, where the cell's internal state begins to shift in ways that take longer to reverse than they took to develop. The cell has reserves, every cell does, glycogen in muscle and liver, small amounts of glucose already inside the cell, already committed to the early steps of glycolysis, ATP already produced and available, a reserve of phosphocreatine in muscle that can regenerate ATP faster than any other mechanism but only briefly. These reserves are not large. Liver glycogen, under fasting conditions, can sustain blood glucose for roughly 12 to 16 hours in a typical adult before it begins to run low. Muscle glycogen is not released into the blood. It exists for the muscle's own use, and it depletes faster during activity than during rest. The reserves exist to buffer short interruptions in supply. They were not designed, if that word applies to anything biological, to sustain a cell indefinitely. When glucose genuinely falls, not to the low end of normal, but to levels where supply is meaningfully insufficient, cells begin making adjustments that reflect a shift in priority. Non-essential processes slow first. Protein synthesis, which consumes a significant fraction of a cell's energy under normal conditions, is among the first to be downregulated when ATP availability drops. The ribosomes continue working but at a reduced rate. The cell produces fewer new proteins. Maintenance gets deferred. The energy that would have gone into synthesis gets redirected toward the processes the cell considers essential. Maintaining ion gradients, preserving membrane integrity, keeping the core of cellular function running at the expense of everything peripheral. This is not catastrophic. Not yet. It is a triage. The cell has been doing versions of this its entire life, adjusting its energy expenditure continuously in response to fluctuating supply. The adjustments made under mild energy stress are reversible. When glucose returns and ATP is restored, protein synthesis resumes, maintenance catches up, the deferred work gets done. The question is how long the deprivation lasts. Prolonged insufficient glucose, the kind that occurs in severe hypoglycemia, or in tissues whose blood supply has been compromised, or in cells that have become so insulin resistant that they cannot take up glucose even when it is present, begins to produce changes that are less quickly reversed. Mitochondria are among the first structures to show the strain. They are the most energetically demanding organelles in the cell, and they are also among the most sensitive to the cellular conditions that develop during energy stress. Rising AMP to ATP ratios, increasing reactive oxygen species, shifts in the redox balance of the cell. Under sustained energy deprivation, mitochondrial membrane potential falls. The organelles that were producing ATP become less efficient at producing it. Some fragment, some are targeted for degradation through a process called mitophagy, cleared by the cell as damaged or non-functional. The cell is consuming parts of itself to manage the deficit. There is something not entirely resolved in how precisely this sequence unfolds across different cell types. Neurons are particularly sensitive. The brain's dependence on a continuous glucose supply means that neuronal energy stress develops faster and with less tolerance for recovery than in most other tissues. Muscle cells have more reserve, more metabolic flexibility, more capacity to shift fuels. Hepatocytes sit somewhere in between. What they share is the basic logic of triage, a hierarchy of functions that get protected and a hierarchy of functions that get deferred, and a point past which deferral accumulates into something the cell cannot fully reverse on its own. The cell does not announce this. There is no outward sign that distinguishes a cell running on depleted reserves from one that is fully supplied. The tissue looks the same. The organ continues functioning. The signs of cellular energy stress are molecular. Enzyme activity patterns, mitochondrial morphology, gene expression profiles, and they require specific measurement to detect. From the outside, the body appears to be managing. Internally, in certain cells, in certain tissues, something has shifted. The work is getting done, but the margin has narrowed. The processes that were running smoothly are running with less redundancy. Small additional stresses, an infection, an increase in demand, a further reduction in supply, will find a cell that has less capacity to absorb them than it had before. It's not entirely clear where the line is between reversible depletion and something more lasting. The research suggests it is not a single line but a gradual transition, different in different tissues, influenced by how quickly the deprivation developed and how long it persisted, and what else was happening in the cellular environment at the time. The cell waited. It adjusted. It deferred. It protected what it could and let go of what it couldn't sustain. And when glucose eventually returned, as it does in most circumstances, before the waiting becomes permanent, the recovery began. Not instantly, not completely in some cases, but the cell resumed, taking up glucose through whatever transporters remained at its surface, rebuilding its ATP, restarting the synthesis it had paused. The reserves refill, the deferred maintenance catches up slowly, and the cell continues, carrying in its molecular state a faint record of what it went through. Altered mitochondrial populations, adjusted enzyme levels, gene expression, patterns that reflect the stress it encountered and the adaptations it made in response. The waiting leaves something behind, not always visible, not always consequential, but present in the cell's chemistry, like a record of a period when the supply was less than the need. And the cell did what cells have always done when that happens. It waited, and it adapted, and it continued. Most people, if asked what insulin does, would say something about sugar, and they would be right, in the way that describing a river by its surface is right, accurate at the level visible from the bank, incomplete in proportion to how deep the water actually runs. Insulin is a pleiotropic hormone. That word, pleiotropic, means it produces multiple effects through a single signal. And for insulin, those effects extend across so many cell types and so many biological processes that the glucose story, as central as it is, occupies only a portion of what the hormone is actually doing at any given moment. Consider protein. When insulin rises after a meal, it does not only respond to the glucose arriving from digested carbohydrate, it responds to the amino acids arriving from digested protein as well. And in response to those amino acids, certain tissues begin doing something quite specific. They increase the rate at which they synthesize new protein. Insulin activates a signaling node called mTORC1, mechanistic target of rapamycin complex 1, which acts as a central regulator of cell growth and protein synthesis, integrating signals from insulin, from amino acid availability, and from cellular energy status. And when all those signals are favorable, mTORC1 promotes the assembly of ribosomes onto messenger RNA and the production of new protein at an accelerated rate. The muscle cell, after a meal, is not only taking up glucose, it is also building. Insulin simultaneously inhibits protein breakdown, a process called proteolysis. By reducing the activity of the ubiquitin proteasome system, the cellular machinery that tags proteins for degradation and dismantles them. Under low insulin conditions, during fasting, proteolysis runs at a higher rate, recycling cellular proteins and releasing amino acids into the blood, which the liver can then use for gluconeogenesis. When insulin rises, this recycling slows. The cell holds on to its proteins. The balance between synthesis and breakdown shifts toward net accumulation. This is why insulin is considered anabolic, not only in the context of glucose and glycogen, but in the literal sense of promoting the construction of biological material from available components. Fat synthesis follows a related logic, and here the story becomes, if you sit with it long enough, genuinely interesting in a way that takes a moment to fully arrive. When glucose enters a liver cell in abundance, more than can be accommodated by glycogen synthesis, more than can be immediately oxidized. The excess carbon does not simply accumulate. It enters a pathway called de novo lipogenesis, where the liver converts glucose-derived acetyl-CoA into fatty acids, assembles those fatty acids into triglycerides, packages them into lipoproteins, and releases them into the blood for delivery to adipose tissue. Insulin drives this entire sequence. It activates the transcription factors, SREBP1C most prominently, that increase the expression of the enzymes responsible for fat synthesis, shifting the liver toward lipogenesis when the post-meal glucose and insulin signal is high. This is not malfunction, it is the body's overflow mechanism, a way of converting excess dietary glucose into a storable form when glycogen capacity has been reached. But it is worth pausing on, because it means that insulin, the hormone most associated with glucose management, is also, under the right circumstances, directly responsible for fat synthesis. The connection between carbohydrate intake and fat storage runs through insulin and through the liver's lipogenic capacity, and it operates as a normal part of post-meal metabolism rather than as a pathological exception. Insulin also reaches into cell survival. Through the PI3K-ACT signaling pathway, the same pathway involved in GALIT. 4. Translocation. Insulin promotes the activity of proteins that inhibit apoptosis, the regulated process of cell death. AC phosphorylates and inactivates pro-apoptotic proteins, reducing the likelihood that a cell receiving adequate insulin will initiate its own destruction. In tissues with high insulin sensitivity, this survival signal is one of the quiet background effects of every post-meal insulin rise, a gentle, continuous reminder to cells that the conditions are favorable, that resources are available, that this is not the time to begin the process of self-dismantling. There is something almost strange in this, thinking about it slowly. The idea that a hormone released in response to a meal is simultaneously telling cells to take up glucose, build protein, synthesize fat, and stay alive all through variations on the same intracellular signaling cascade, all because a cluster of cells in the pancreas detected that glucose in the blood had risen above a threshold. One signal, many instructions. The cell receives all of them at once and somehow integrates them, running glucose uptake alongside protein synthesis, alongside lipogenesis, alongside survival signaling, allocating its molecular machinery across all these tasks simultaneously, prioritizing according to its own type and state and history. Insulin does not coordinate this integration, it initiates it. what happens after the signal arrives, which instructions get followed most fully, which pathways run most actively, how the cell balances competing demands on its energy and synthetic capacity, is determined by the cell itself, by its current state, by what it was doing before the signal arrived, and what it has the machinery to do in response. The hormone speaks once, the cell decides what it heard, and somewhere downstream of that decision, in the quiet interior of a muscle cell or a hepatocyte or a fat cell, the work of acting on the instruction, building, storing, surviving, continuing, unfolds without announcement, in the same unhurried way that most of the body's most consequential processes have always unfolded. Deeper than the glucose story, still running, there is a protein on the surface of your cells that has been responding to insulin since before you were born. Not the same protein molecule. Proteins don't last that long, most of them turning over within days or weeks, degraded and replaced continuously throughout the life of the cell. But the receptor, as a structure, as a design, has been present in your cells since the earliest weeks of embryonic development, expressed in tissues that were forming before you had the metabolic needs that would eventually make the receptor's function essential. The insulin receptor is a tetramer. 2 alpha subunits sitting outside the cell, available to bind insulin, joined to 2 beta subunits that span the membrane and extend into the cell's interior, where they carry the enzymatic machinery that initiates signaling. When insulin binds to the alpha subunits, the receptor undergoes a conformational change, a physical rearrangement that activates the tyrosine kinase domains on the beta subunits, which then phosphorylate each other and begin the cascade that eventually reaches IRS proteins, PI3K, AC8, and everything downstream. This much is relatively settled. What is less often considered is what happens to the receptor after it fires. Repeated activation changes the receptor's availability at the cell surface. When insulin binds and the receptor is internalized, pulled inward through a process called receptor-mediated endocytosis, enclosed in a vesicle and drawn into the cell's interior, it enters a sorting process that determines its fate. Some receptors are recycled back to the surface, others are targeted for degradation. The ratio between recycling and degradation is not fixed. It shifts depending on how frequently the receptor has been activated, how much insulin has been present, how long the signal has been sustained. A cell chronically exposed to high insulin concentrations ends up with fewer receptors at its surface. This is called downregulation. It is not, you start to notice, entirely different from what happens in other sensory systems, the way a sound that was startling becomes unremarkable after repetition, the way a smell that was vivid on entry fades within minutes of sustained exposure. The receptor system is doing something analogous, though through entirely molecular means. It reduces its own availability in proportion to how much it has been used. The cell is, in this limited sense, adjusting its sensitivity based on its history. What came before shapes what the receptor can do now. A cell that has been receiving strong insulin signals for months or years has, through the ordinary process of receptor regulation, reduced the number of receptors available at its surface. Less surface receptor means less signal per unit of insulin. Less signal means less downstream activation. Less activation means a blunted biological response, not because the receptor is broken, not because the insulin is defective, but because the cell has done exactly what receptor regulation is designed to do, and in doing it under conditions of chronic excess, has arrived at a state that functions like resistance. The past is present in the receptor count. There is a related phenomenon at the level of the signaling proteins downstream. Repeated activation of the insulin signaling pathway leads to feedback phosphorylation events. IRS1 being phosphorylated on serine residues by kinases activated by the very pathway that IRS1 belongs to, a form of negative feedback that reduces the signal's amplitude with each successive activation. This too is a form of cellular memory, not memory in any conscious sense, but a record of recent activity encoded in the phosphorylation state of proteins that shapes how the next signal is received. The receptor fires, the cascade runs, the cascade activates kinases that phosphorylate IRS1 in an inhibitory way. The next time insulin arrives, the pathway is slightly less responsive than it was before because the previous activation left its mark. Under normal conditions, this negative feedback is useful. It prevents the cell from over-responding to sustained or repeated signaling. It is part of the regulatory architecture that keeps the insulin response proportional and controllable under conditions of chronic overstimulation. Well, the feedback that was designed to fine-tune the response becomes, over time, a source of sustained attenuation. The serine phosphorylation that was meant to be transient becomes more persistent. The receptor downregulation that was meant to be reversible becomes a new baseline. The cell's sensitivity to insulin settles at a lower level than it was at before, and the pancreas, detecting the blunted response through the blood glucose signal it monitors continuously, increases its output to compensate, which sustains the high insulin environment, which maintains the receptor downregulation and the IRS1 inhibition, which keeps the sensitivity low. The loop, running quietly, each element reinforcing the others, not initiated by any single failure but emerging from the intersection of normal regulatory mechanisms operating under conditions they were not originally tuned for. What you start to notice sitting with this is that insulin resistance as a cellular phenomenon is not really a malfunction in the conventional sense. The receptor is working, the feedback systems are working, the downregulation is working, everything is doing what it is supposed to do. The problem, if it can be called that, is that the conditions have changed. The sustained elevation of insulin, the chronic glucose load, the inflammatory environment, and the regulatory systems that were shaped for a different kind of environment are producing an outcome that those conditions never required them to navigate. The receptor adapted to what it was given, and what it was given over time shaped what it became, not through any failure of design, but through the ordinary operation of mechanisms that have always been there, responding to history the only way biology knows how to respond to anything. By adjusting, by changing the present in proportion to what the past contained. The receptor on your cell surface right now carries that history in its count, in its phosphorylation state, in the ratio of recycled to degraded copies that determined how many ended up available this morning. It doesn't remember in any way that resembles memory, but the past is in it, quietly, the way the past is in everything biological that has been running long enough to have one. There is no single place where the body tallies what insulin has done. No organ sits above the system, reviewing the results, deciding whether the response was adequate. The accounting happens everywhere at once, in the concentration of glucose remaining in the blood, in the rate at which the liver is still releasing or has stopped releasing, in the phosphorylation states of enzymes in muscle cells that reflect how fully they responded to the signal they received. The score is distributed. It exists only as the sum of states across tissues, readable only by the system itself, and only in the language of molecular concentrations and enzyme activities rather than numbers on a page. This is worth staying with for a moment. The body's feedback loops, the mechanisms by which it detects the outcome of what insulin has done and adjusts its next action accordingly, are not centralized in any control structure. They are embedded in the tissues themselves, running in parallel, each loop operating on its own timescale and responding to its own set of signals, the collective behavior of all of them producing something that functions like regulation without any single regulator overseeing the whole. The pancreas runs its own loop, and it is the most immediate. Beta cells monitor blood glucose continuously, and as insulin drives glucose into tissues and blood glucose falls, the beta cells detect the decline and reduce their secretion in proportion. This feedback operates on a timescale of minutes, the same timescale as the initial glucose rise, making it fast enough to prevent insulin from driving glucose too low, responsive enough to track the curve of post-meal glucose as it peaks and begins its descent. The loop is elegant in its simplicity. More glucose drives more insulin, less glucose drives less, and the relationship between the two is graded and continuous rather than binary. But the pancreatic loop alone is not sufficient to explain the precision of glucose regulation. The liver contributes its own loop, operating on a slightly different timescale and responding to a different set of signals. As blood glucose falls and insulin declines, glucagon, secreted by alpha cells in the same pancreatic islets that contain the beta cells, begins to rise. Glucagon reaches the liver and activates glycogen phosphorylase, the enzyme that breaks down glycogen and releases glucose back into the blood. The liver's output increases in proportion to how far glucose has fallen and how much glucagon it is receiving, providing a counter-regulatory force that prevents the insulin-driven decline from continuing unchecked. Glucagon and insulin are not simply opposites. They are participants in a dialogue that the liver mediates, each modulating the other's effect, each shaping what the liver does with the glucose it holds in storage. The ratio between them, not the absolute level of either, is what the liver reads most sensitively, adjusting its net glucose output according to which signal is currently dominant and by how much. You already know this balance exists, in some bodily sense, even if you have never had reason to think about it explicitly. The feeling of blood glucose stabilizing after a meal, the return of clarity after the post-meal softness, the quiet restoration of baseline energy, is partly this loop completing its work, the liver resuming its output at a rate calibrated to replace what the tissues are consuming, the glucagon signal having risen just enough to keep the decline from going further than it needed to go. Muscle participates in the accounting differently. Skeletal muscle does not release glucose back into the blood. It lacks the enzyme glucose 6-phosphatase that would allow it to do so, but it adjusts its rate of glucose uptake as insulin falls, drawing less from the blood as the post-meal signal fades and shifting toward fat oxidation as fatty acid availability increases. In this way, muscle contributes to the score not by adding glucose back but by reducing its withdrawal, releasing pressure on the supply at the same rate that the liver is restoring it, the two adjustments running in parallel and in the same direction without any direct communication between them. They are responding to the same signals, arriving at compatible outcomes. Without coordination, this is the part that tends to stay with you. The idea that a system this precise, this responsive, this capable of maintaining blood glucose within a relatively narrow range across an enormous variety of inputs and demands achieves its precision not through central control, but through the distributed behavior of tissues, each running their own feedback, each responding to the same circulating signals, each contributing a portion of the regulatory work that collectively adds up to something that looks, from the outside, like a well-managed system. It is a well-managed system. The management is just not located anywhere in particular. Adipose tissue completes its own part of the accounting over a longer time scale, releasing fatty acids as insulin falls and gradually restoring the fuel mix that the body uses during fasting. The slow resumption of lipolysis, adding a third fuel stream to the post-meal recovery, alongside hepatic glucose output and reduced muscle uptake. The accounting in fat tissue runs over hours rather than minutes, its contribution to the score accumulating slowly, becoming most visible in the late post-meal period when insulin has been low long enough for fat release to be fully restored. And through all of it, through the rapid pancreatic loop, the hepatic counter-regulation, the muscles shifting uptake, the fat tissue's gradual reopening, blood glucose traces a curve that begins rising with the meal, peaks sometime in the first hour, and returns toward baseline over the following 2 to 3 hours, the exact shape of the curve reflecting the sum of all these loops running simultaneously, each contributing its portion of the result. The body keeps score the way all distributed systems do, not perfectly, not always, but continuously, and with a resilience that comes from having no single point of failure, from the accounting being spread across so many tissues, so many loops, so many overlapping mechanisms, that the failure of any one of them can, for a time, be absorbed by the others adjusting to compensate. The score is never final. The next meal is already being anticipated, somewhere in the beta cells monitoring the blood, somewhere in the liver holding its glycogen, somewhere in the fat cells that have just finished reopening and are already, quietly, beginning to prepare for the next time they will be asked to close. Insulin leaves quietly, not all at once, not with any announcement that the post-meal phase has ended and something else has begun. It clears from the blood gradually, carried to the liver and kidneys where enzymes dismantle it, its half-life in circulation somewhere between 4 and 6 minutes, which means that within 20 to 30 minutes of the pancreas reducing its output, most of what was released after the meal has already been degraded. The signal is brief, what it leaves behind is not. The GLUT4 transporters that migrated to the surface of muscle and fat cells during the insulin peak do not immediately return to their intracellular vesicles when insulin begins to fall. They linger. The machinery that retrieves them, clathrin-coated pits forming at the membrane, vesicles pinching inward, the internalization process that reverses what insulin triggered, operates on a timescale that lags behind the hormone's clearance. For a period of perhaps 30 to 60 minutes after insulin has already declined significantly, transporters remain at the surface in numbers that still exceed the pre-meal baseline. Glucose continues entering. Slower than before, the gradient has narrowed, but the gates are still there, still open, still admitting what they can from a blood supply whose glucose concentration is already approaching its fasting level. The cell is responding to a signal that has mostly passed. This is not an error. It is a feature of how receptor systems work, a built-in persistence that extends the biological effect of a transient signal, smoothing the transition between states rather than producing an abrupt switch. The body moves between post-meal and fasting physiology the way light moves between day and dusk, and gradually with an interval where both are partially true. Inside the muscle cell, the work that insulin initiated is still running. Glycogen synthase, activated during the insulin peak, continues adding glucose units to glycogen chains even as the signal driving its activation fades. The enzyme's active state persists past the signal's peak, maintained by the phosphorylation changes that insulin triggered, which take time to reverse through the phosphatases that gradually restore the enzyme to its baseline activity. Glycogen continues accumulating quietly in the minutes and hours after the meal, the cell completing a storage process that insulin started but did not need to stay present to finish. The instructions were given. The work continues without the instructor. Something similar happens in the liver. The gene expression changes that insulin promoted, the increased transcription of enzymes involved in glycolysis and lipogenesis, the decreased transcription of gluconeogenic enzymes, persist beyond the hormone's clearance because gene expression changes operate on timescales measured in hours, not minutes. The messenger RNAs that were produced during the insulin peak are still being translated into protein long after insulin itself has gone. The liver's metabolic posture, shifted by the post-meal insulin signal, remains shifted for a period that extends considerably past the signal's duration. The hormone spoke once, the cell listened for longer. You start to notice, sitting with this, that the concept of insulin's effect having a duration is more complicated than the concept of insulin having a half-life. The half-life describes how long the molecule persists in the blood. The effect's duration depends on which effect you are measuring: transporter localization, enzyme activation state, gene expression, glycogen accumulation, and each of these has its own timescale, its own rate of return to baseline, its own persistence past the signal that initiated it. Some effects last minutes past the signal. Some last hours. Some, the receptor downregulation, the altered gene expression patterns in chronically stimulated cells, the accumulated serine phosphorylation on IRS proteins, persist across days or longer, carrying the history of past insulin exposure into the cell's present state in ways that shape how it will respond to the next signal when it arrives. The meal ends, insulin clears, and inside the cells that received the signal, the muscle holding its newly built glycogen, the fat cell closed and full, the liver running the expression patterns that the post-meal insulin established. The work of the meal continues quietly and without the hormone that started it. Longer than the hormone itself was present, this is what remains. Not the signal, not the molecule, but the changed state of the tissues that received it. Altered enzyme activities, repositioned transporters gradually returning, gene expression patterns still running out their course, glycogen stores fuller than they were before, the body in a different configuration than it occupied this morning, carrying in its molecular state the record of everything that has passed through it today. By the time you are still enough to notice the quiet, the breath slowing, the body settling, the particular kind of rest that comes after a long day of metabolic work, the insulin from your last meal has long since been cleared, dismantled, gone. But what it changed is still here, still running, the transporters almost back, the glycogen sitting full and still in the muscle, the liver, waiting for the hours ahead when the body will need to draw on it again, when blood glucose will fall and glucagon will rise, and the stored glucose will leave its chains and enter the blood and travel outward to the tissues that are waiting, as they are always waiting, for what the body has set aside for them. The signal fades, the body it changed remains, and somewhere in the pancreas, in the islets that have been measuring and releasing, and measuring again since long before this day began, the beta cells continue their quiet work, monitoring the blood, reading the concentration, waiting for the next threshold to be crossed, as they have always done, as they will continue doing tonight while you sleep. If you made it here all the way through, then you already know what this channel is. The sleepy biologist exists because biology is most beautiful when it is unhurried, when it has room to unfold at its own pace without urgency, without the pressure of a quiz at the end or a highlight reel to get through. If this video found you at the right moment, if something in it settled you or quietly surprised you or gave your mind something gentle to hold while the rest of you let go, then consider leaving a like. It takes a moment, and it helps more than you might expect. If you want to be here for the next one, subscribing ensures you will be. There is always another system running quietly inside you, waiting to be looked at slowly. For those who support the channel through membership or super thanks, thank you. That support is what makes the long format possible. It is what makes the unhurried version possible. If you have ever sent a super thanks or held a membership, you are part of why this exists. Leave a comment if you'd like, where you listened from, what stayed with you, what you're curious about next. Those comments are read. They matter to what gets made. Rest well. There is more biology waiting for you whenever you are ready.